What if you could go to a dentist and receive a treatment that would allow your tooth to heal itself rather than dealing with a dental drill? We are on our way there. And in fact, we've got some really cool options available to us right now at dentist offices. They're just a little bit lesser known. Dentistry is shifting away from drilling and towards biological repair. So let's just go over the anatomy really quick, and then we'll cover the details, talk about what's in the clinic right now, and then also talk about what's promising in R&D. You've got the hard outer layer of enamel, and then you've got a layer underneath called the dentin, which supports the enamel. It absorbs the stress of chewing, protects the inner pulp, and it also acts as a sensory network. So it transmits sensations like temperature and pressure to the pulp, but then eventually to the brain. So at this point, the science is telling us that not only are we able to stop a cavity dead in its tracks, but we're also able to remineralize early enamel lesions. And so these are just white spots on your teeth that are about to develop into a full-blown cavity. In the longer term, we might also be able to stimulate dentin to repair itself when the damage is a little bit deeper. In this video, we are going to cover some of the leading breakthroughs in tooth and cavity regeneration. So we'll start with Curodont and the P11-4 self-assembling peptide. We have Silver Diamine Fluoride, Tideglusib, or NP928 in its current form. We've also got enamel organoids, and last but not least, this one's hot off the presses, keratin-based enamel repair using materials derived from wool. Let's start with Curodont, which is actually on the market right now, and it deals with damage to the enamel. Its active ingredient is a self-assembling peptide called P11-4. Now, peptides are just shorter, smaller proteins. And in fact, you can think of proteins as being made up of a bunch of Lego pieces, and we call these amino acids. So a peptide is anything from 2 to 50 Lego pieces, while a full-blown protein would be anything above 50 Lego pieces. When applied to an early non-cavitated lesion, so again, that's just a chalky white spot on your tooth that's likely to develop into a cavity, it'll seep into the porous enamel and it'll self-assemble into a 3D matrix. That 3D matrix will attract calcium and phosphate ions from your own saliva to remineralize the enamel. I just think that's so cool. You literally have all of the pieces required in your saliva to make this work post-application. These minerals will crystallize into hydroxyapatite, and that's the same substance that your enamel is made of. However, I do just want to note that this is not classical regeneration in the sense that I'm going to give a drug and the tissue is going to heal itself and build back. No, this is remineralization. So all I'm doing is providing a scaffold to pull minerals back into the tooth from an external source and build that barrier back up again. There are clinical trials that have shown that P11-4 can reverse early enamel lesions, and it outperforms fluoride varnish, which is also used to remineralize teeth. Plus, a 2023 systematic review and meta-analysis found that P11-4 significantly improved the arrest of cavity formation and reduced progression relative to fluoride alone. When you combine the two treatments together, as they've done in Curodont Repair plus fluoride, the results are even stronger. On the practical side, this treatment can be applied in just a few minutes, and per the manufacturer, it doesn't stain, which means it can be used on the front teeth as well as back teeth. Curodont is currently being marketed for professional use in the US, and it's been available in Europe for years. So, if you're prone to cavities, or if you've recently had orthodontic treatment, and now you're dealing with demineralization issues, you've got those white spots on your teeth, this is definitely worth asking your dentist about. The second major breakthrough that we're covering today is Silver Diamine Fluoride, or SDF. SDF is FDA cleared to treat tooth sensitivity, so of course, that's just when you eat or you drink something hot or cold and then your teeth hurt. But now it's being used off-label to arrest cavities, stop them in their tracks. SDF works by combining silver ions, which kill decay-causing bacteria, with fluoride, which, as we mentioned before, remineralizes the enamel. So in effect, what you get with that combination is that you're hardening the softened tooth structure, but then you're also halting further decay. When SDF is repeatedly applied to an early cavity, you can get really high arrest rates. We're talking 75 to 90% in many studies, especially in primary teeth. The trade-off here is that you will get black staining at the site of the cavity. That makes it maybe not the best option for front teeth, far more often used on those back teeth that you can't really see so well. So that being said, it's got an excellent safety profile, highly effective, Definitely worth considering. Now let's move deeper into the tooth beneath the enamel where we find the dentin. Damage to the dentin has traditionally required fillings, but researchers are currently taking a totally different approach. Enter tideglusib, a drug that was originally developed for Alzheimer's disease. Tideglusib inhibits an enzyme called GSK3. And this in turn activates the Wnt signaling pathway, one of biology's master regulators of stem cell behavior and tissue regeneration. In rodent studies led by Dr. Paul Sharp's group at King's College London, researchers created small holes in the animal's teeth. Now these holes extended past the enamel and down into the dentin. What they then did was they placed tiny biodegradable sponges soaked in tideglusib into these holes. Over several weeks, these sponges would break down, and the tooth's own stem cells would be activated to form new layers of dentin and restore that internal structure. These experiments prove that the teeth can regenerate from within given the right biochemical signal. A next-generation compound called NP928 works much the same way. It's basically just a version of tideglusib that was structurally tweaked to become more water-soluble, and that way it can be delivered as a gel instead of a sponge. Much easier to work with in real-life dental settings. NP928 has not been tested in humans yet. However, it is highly similar to tideglusib in terms of its structure. And tideglusib has been through multiple human clinical trials at this point for an array of neurological conditions. So we're optimistic about the safety profile of NP928. Still, it needs to be tested in its own human clinical trials for the indication that we're interested in. So that would be for dental applications specifically. I actually had a quick email exchange with Dr. Paul Sharp, and he told me that they're just having a really hard time finding funding for the clinical trial. And that's despite the fact that it's all planned out and that they're talking to the relevant regulatory bodies for clearance. So as of this moment, there are no human dental clinical trials that have been registered or published for NP928 or tideglusib. Hopefully that changes soon. Either way, tideglusib and NP928 look to be among the most promising routes toward dentin regeneration using your own stem cells. The next innovation pushes even further into the realm of regenerative biology, but this time, instead of dealing with the dentin, we are back up at the enamel. Researchers have successfully created enamel organoids. So these are tiny 3D structures grown from stem cells that mimic the way enamel forms in the body. And so I actually have a whole video on organoids. If you're interested in watching it, you can click on the card above. Published work has shown that enamel organoids can secrete the right proteins and make the right minerals in vitro. In vitro just means in a dish. That would contrast in vivo, meaning within the body of a living thing. I've got a short on that as well. Link is in the description. So these findings are limited to the preclinical category. We haven't really shown that this works in humans yet, but we will keep an eye on these findings. In the future, this work could allow for the creation of bioengineered enamel patches or caps to be applied to areas where tooth is damaged. But all of this is just theoretical so far. Nothing's actually been proven yet. So much stunning work has been done using stem cells in the clinical setting to date. So I'm sure that this is going somewhere useful, even if we're still in the preclinical phase of study. This last one is hot off the presses, published August 2025. Scientists have found a way to use keratin, the same substance that's found in human hair and human nails, to repair tooth enamel. In their experiments, the researchers sourced their keratin from wool, which is a great idea because sheep produce so much of it and they needed large volumes for their experiments. They engineered their keratin into tiny nanostructures that resemble the crystalline arrangement of enamel. They then applied this material to etched or demineralized human enamel, and what they found was the keratin was able to bond to the tooth surface and form a scaffold for mineral deposition. So this is very similar to what I described earlier. Since keratin is a natural, biocompatible protein, this approach could one day lead to enamel-like coatings. Some people have even been talking about toothpaste. However, none of that has been tested clinically yet. This discovery links to seemingly unrelated biological materials, hair and teeth, very cool. And one of you guys actually left a comment making this connection on my last tooth regeneration video. I don't know if this person realized how right on they were. I'm not sure if they saw the study or what exactly the deal was. Either way, this is why I love going through your comments. Together, these technologies are transforming dentistry from being very much centered around mechanical restoration to focusing in on biological repair and regeneration. Very exciting stuff. So if you learned something, share this video with a friend.